Okay, I think we'll get started. Dear participants, my name is Gavin McDonough, and I'm the Educational Action Supporting Officer at ERN-EuroBloodNet, the European Reference Network for Rare Hematological Diseases. And it is my great pleasure to welcome to ERN-EuroBloodNet Topic on Focus on Constitutional Thrombocytopenia, which is an accredited European online educational program targeting health professionals organized by the ERN-EuroBloodNet and in partnership with the CRPP, French Reference Center for Inherited Platelet Disorders, and the Filière MHEMO, which is the French Constitutional Bleeding Disorders Network. The program consists of a cycle of webinars focused on constitutional thrombocytopenia, developed in collaboration with three experts, Professor Marie-Christine Alessi and Dr. Paul Saultier from Assistance Publique-Hôpitaux de Marseille, and they're also members of the ERN, and Dr. Mathieu Fiore from CHU Bordeaux, both of which are here in the webinar today. But before we start, let me share some home rules. Please note that the session is being recorded and will be uploaded to the EBN e-learning environment. If you do not wish to appear, you can turn off your camera and change your name on the platform. And also, please be kindly informed that your microphone is muted. If you have any questions, please put them in the chat, and there will be a small or short Q&A session at the end of the webinar. The program is accredited with continuing medical education points provided by the European Board for Accreditation in Hematology, and those credits are assigned to the participants of a single session, so 1 credit, and those that participate to the full program of the 15 webinars can get 15 credits. There will be a short survey at the end of the webinar. Please use the same name and last name and email that you used to register for this webinar. But with that, I have the pleasure today of introducing the webinar by Dr. Kathleen Freson on the benefits and limitations of high-throughput sequencing for the diagnosis on constitutional thrombocytopenia. Dr. Kathleen Freson is a professor at the University of Leuven in the Department of Cardiovascular Sciences and the head of the Center for Molecular and Vascular Biology. She is co-chair of the ISTH subcommittee of genomics and thrombosis and hemostasis, elected vice chair of the SSC from ISTH, and a member of the NIH-funded ClinGen Working Group for Thrombosis and Hemostasis. And with that, it is my pleasure to provide you the floor. Thank you very much for joining us today.
Thank you very much for the kind invitation. Also, I would like to thank the organizers of this webinar series for giving me the opportunity to present some of the work done on high-throughput sequencing. Okay, this is my only disclosure for this presentation. For the webinar, I have 3 learning objectives. So I would like to introduce the gene panel test and how it is best designed to diagnose inherited thrombocytopenias. I would also give you an overview of what you can expect in terms of diagnostic rates when you use this to diagnose patients with inherited thrombocytopenias. And then I also would like to discuss the pros and cons of using an NGS panel test and thereby also give some examples from the clinic. So when we use an NGS panel test for inherited thrombocytopenias, the first thing is how do you design an NGS panel test, and therefore it is important to pick the right genes, and these are called diagnostic rate genes that should be included on a gene panel test. In this schedule, you see genes that we currently know cause inherited thrombocytopenia, and these genes are all important in different aspects that regulate megakaryopoiesis and platelet production. The genes, as shown here, are clustered based on their predicted role that they have in megakaryopoiesis. So, for instance, you have genes that regulate transcription factors. You also have genes, that are important for glycoprotein signaling, genes for the cytoskeletal regulation. And then there are several genes where we don't know their specific role in megakaryopoiesis. So these genes are not randomly chosen, but these genes have been created over the last years. And the creation work for genes that are important for inherited thrombocytopenia has been part of the ICTHSC subcommittee for thrombosis and hemostasis. There's a paper in GTH that describes this gene creation work and how this was done. Briefly, I can mention that genes that have been created are known to cause inherited thrombocytopenia. Yearly, we get updates of genes because new genes are discovered over the years, and so we add new genes to this list every year. In addition to that, there is a second organization that has taken up some gene creation work. So I don't know why my slides are automatically progressing. So the ClinGen group has, there is a ClinGen group that is specifically looking at genes that are involved in thrombosis and hemostasis. And also they have taken up work to create genes, but the gene lists overlap between these two organizations. This slide specifically gives an overview of genes that cause thrombocytopenia, and they are divided in their effect on either creation large or small platelets or normal sized platelets. And in addition to that, I have grouped the genes that cause just only thrombocytopenia, but as is probably known by this public, many of the genes also cause syndromic forms of thrombocytopenia. And you see that there are different genes that, for instance, cause large platelets in addition with a syndromic form of thrombocytopenia. And specifically for this reason, we have developed NGS panels, not to make making sure that you can screen all the genes in a single step, and therefore no need to screen gene by gene. So in the past, we have used Sanger sequencing and MPL or copy number variant analysis on a gene by gene order to diagnose patients with inherited thrombocytopenia. Nowadays, we use NGS panel tests that either are targeted or can use exome or genome sequencing to diagnose patients with inherited thrombocytopenia. The first attempt to use an NGS panel test was done by the thrombogenomics study that started in 2016. This was an international study that was coordinated in Cambridge, and the purpose was to see whether we can use an NGS panel test to diagnose patients with inherited platelet disorders. The first study by the thrombogenomics was just a test whether it was feasible to do NGS screening using a panel test, and this panel test included not only genes that are relevant for thrombocytopenia, but also genes that cause thrombosis and bleeding pathologies. The outcome was that this targeted panel test was very accurate and had a 99 to 98% coverage, which is extremely high, meaning that you cover most of the genes that are on the panel test, except for some small regions that need to be filled with Sanger sequencing. In addition to that, the lesions and other types, so insertions could be detected, except for gene inversions. So if you, the only type of the variants that you cannot pick are gene inversions. Also, very important is that when you screen a patient for all the genes that cause bleeding and thrombosis, you find a mean of 5 variants per individual, and that has important consequences. And I will give you some examples of that during my talk. Associated with this is that you need to go over the variants one by one to discuss whether they're clinically relevant or not, because not all variants that you find are of clinical relevance. And then a gene panel test is affordable. By multiplexing samples, the sequencing cost has dropped enormously, and therefore the NGS panel test has reached the clinic in many countries. So this then resulted a few years later in the actual application of the gene panel test for bleeding and thrombosis. And I specifically want to draw your attention today to the diagnostic rate that we could obtain when screening patients with inherited thrombocytopenia. Nearly 50% of all the patients got back a report with a variant that could explain their clinical phenotype. Now, all the variants were classified using the criteria that were developed by ACMG, and all the variants were classified into pathogenic, likely pathogenic, or variant of unknown clinical significance. The difference is that pathogenic and likely pathogenic variants do cause the disease, while for variants of unknown clinical significance, we don't know whether they're clinical relevant or not. Typically, variant of unknown clinical significance should not go back to patients or should not inform clinical management. So you see in the patients with inherited thrombocytopenia, which involved 335 patients, a significant portion here in green still were explained by a variant of unknown clinical significance. And this is the main problem that we have with NGS panel tests that we use today in the clinic, and not only in our field, but in all other rare diseases. We find a significant portion of variants that we cannot yet use to inform clinical management. So in this slide, you see the variants and the genes that were found specifically in the group of patients with inherited thrombocytopenia. You see the most common gene where we found variants was MHEMO, and then you see the order here listed on the variants, but a significant portion of patients with inherited thrombocytopenia, though we screened 300 patients, were unique cases. So most genes that cause inherited thrombocytopenia are extremely rare. So where does this lead? The thrombogenomics panel, as it was used between 2016 and 2019, was still a research project. So after 2019, we have implemented a similar NGS panel test for bleeding and thrombosis in a clinic here in Belgium. So we recruit at this moment nearly 200 patients yearly. And these involve children and adults in equal proportion. And we do a similar screening as done for the thrombogenomics panel, with the difference that we use exome sequencing and not targeted sequencing. An interim analysis was published some years ago, and Christine van Laar is the PhD student in my group who leads this panel. Starting in 2019 till today, we have almost screened 1,000 patients by this panel, and they have been recruited all over Belgium because we are the referral center for genetic testing in Belgium. So how does this work? Upon inclusion, we ask clinicians for an informed consent, clinical data, and a DNA or a blood sample to send to Leuven. Based on these data, processing of the samples is done at the Center of Human Genetics. They do it simultaneously for all the rare diseases. Variant processing, variant filtering is done by the Human Genetics Lab, and variants together with clinical data are processed in a software package that's called Alessi from Illumina. Afterwards, I look at the data together with different team members. There is a member from the Center of Human Genetics, Christine van Laar, as head of the clinical biology. And we invite the clinicians to go over the genetic data and discuss the variants one by one. When the variants reach the level of pathogenic or likely pathogenic, they are taken up in the clinical report and they are fed back to the patients to inform their clinical management. Variants of unknown clinical significance are not fed back in the clinical reports, but are pushed through the diagnostic line to the research lab to perform additional testing. For variants of unknown clinical significance, we recall patients to take other blood samples, and we invite family members to participate in variant co-segregation analysis. This to upgrade variants of unknown clinical significance when they co-segregate or when we find specific functional assays that are normal, and we classify the variants into likely pathogenic to be able to use these variants in a clinical workflow. As other tests, we use a similar variant reclassification schedule as I discussed before, and that is used by most labs based on the ACMG criteria. So what we have as outcome today, so we screened about 1,000 patients. Both are referrals from the clinic and from the adult centers. When we look specifically to the panel that is mostly covering the inherited thrombocytopenias, this here indicated in orange, we have a similar referral from the adults and clinicians, and 30% of all referrals have to do with inherited thrombocytopenia. When we look at the diagnostic rate, including variants of unknown clinical significance, we have about a 50% diagnostic rate. When we again split the discoveries all over the genes, most discoveries cover the Glanzmann thrombosthenia gene IgG2B, where we do find some genes that cause thrombocytopenia. In addition, we have genes that are causing Bernard-Soulier syndrome, MIH9, tubulin actinin, and so on. Now, what you also see on the slide here is that we have van Willebrand factor on our panel test for inherited thrombocytopenia. And what's the meaning of that is that we find in a significant portion of the gene a pattern that's called degenic inheritance. So we do find variants in van Willebrand gene as modifier for bleeding in patients with inherited thrombocytopenia, meaning that patients with inherited thrombocytopenia have a gene that causes thrombocytopenia, but often also have a variant in the van Willebrand gene, typically associated with type 1 van Willebrand, that modifies their risk for bleeding. In addition, as you see here in yellow, many patients do still contain variants of unknown clinical significance in genes that cause thrombocytopenia, and this is a significant burden for patients and actually the diagnostic rate that we see today. What are pros and cons of using an NGS panel test? I don't go over one by one, as I just explained the most important ones. The strength is actually that this is an affordable way to screen your patients. And I will show you some examples later. You find unexpected diagnosis. And I will give you an example what I mean with that in a few minutes. What are the most important limitations? Already named the most important ones. This is the high discovery of variants of unknown clinical significance, where you don't know what to do with today. In addition to that, there are some ethical concerns, specifically when you screen for inherited thrombocytopenias, and I will give an example in the next slides. So what can we do to reclassify variants of unknown clinical significance? This was a project called Gold Variants, was initiated by the SSC for Genomics and Thrombosis and Hemostasis some years ago. So what we actually want to do with the gold variants is that this is a variant capture tool. So we want to reach out to all the diagnostic labs that use a gene panel test and capture variants of unknown clinical significance all over the world. At this moment, we have reached out to 30 diagnostic labs from 14 countries, and we have captured at this moment almost 1,500 variants. And you see that in all the diagnostic labs, a significant portion of 40% of all the variants that they pick up in the NGS panel test or variants of unknown clinical significance. What is the purpose now? We want to capture these variants and look whether we see similar variants occurring in patients with similar genotype-phenotype correlations. That would give us the opportunity to upgrade these variants of unknown clinical significance and reclassify them into a likely pathogenic status before uploading them into ClinVar, which is the variants database that is typically used by diagnostic labs to classify their variants. Another important shortcoming is what I mentioned is the ethical implications that we have to deal with when we use NGS testing for inherited thrombocytopenias. It's known that you have a risk to find variants that were not a primary rationale for testing. And in our field, variants that were not the rationale for testing are, of course, variants that we can detect when we screen patients with inherited thrombocytopenia in RUNX1, ETV6, and anchorin RD26. Variants in these genes can cause a risk factor for leukemia. And we believe that it is very important to inform patients upfront of doing the genetic test that these variants can be found, allowing them or giving them the opportunity to opt out when they don't want to know this before doing the tests. I give an example of that to give more idea what this means. So in our gene panel test in Leuven, we have done a test for a patient who had mucocutaneous mild bleeding symptoms. She had a platelet count that was in the lower range, but on some occasions the platelet count was normal. And the main phenotype of this patient was actually that she suffered from platelet delta storage pool disease with low ATP levels. Now, before doing the test, this patient gave informed consent and actually agreed to have the full panel test done. The outcome of the panel test for this patient was that we found a pathogenic variant in the RUNX1 gene. And this was, of course, a very unlikely finding because she had a rather normal platelet count. However, it's known that pathogenic variants in both LY1 and RUNX1 can cause relatively normal platelet count numbers and are often associated with platelet-dense granule secretion defects. Now, for this family, we follow them up very closely in the clinic, and actually this family is very cooperative, and most members in this family want to know whether they're carrier of this RUNX1 pathogenic variant. You see that we could recall several members of this family, and you see that most of the members who have the pathogenic variant have relatively normal platelet counts or in the lower range. Some years ago at the EAT meeting, I got the opportunity to do a poll during the meeting, and I actually had two important questions asked to the public. And the first question was whether the participants during that meeting, whether they use a gene panel test to diagnose patients with thrombocytopenia, and whether they told their patients before doing the test whether they wanted to know whether they would, had variants in genes that cause risk factor of leukemia. You see here the outcome of the panel is that only 50% of the people who do the panel test inform their patients before the onset of the test. Another question is that whether they take an informed consent before doing the test. 60% do, 40% do not. So you see that at the global scale, there is no real, that everybody, or there's no real agreement on whether we should do this or not. And there is no uniform policy related to this type of testing. Another important actually, I think a good thing to do is that you can use an NGS panel test to diagnose patients where you would miss the diagnosis just based on classic clinical diagnosis and doing lab assays. So I give again an example. This is about a patient from 22 years. She was suffering from bleeding after trauma and menorrhagia. You see she has microthrombocytopenia with very large platelets. Platelet aggregations were normal and ATP secretion as well. FACS showed that CD61 and 41 were normal, and CD42 had a 50% expression level. The most profound lab abnormality was the prolonged PFA time. When we screened this patient, we found a dominant form of Bernard-Soulier syndrome, and we found second variant where we don't know whether it's relevant or not. She also had a missense variant in the tubulin 1 gene. The glycoprotein 1BB gene for dominant Bernard-Soulier syndrome was a known pathogenic variant. We believe, however, that two variants play an important role here in the phenotype of this patient. It's known that the dominant form of Bernard-Soulier syndrome given in between phenotype with the classic Bernard-Soulier syndrome, enlarging platelets and generating a mild reduction in the platelet count. However, we also saw that this specific variant in tubulin was described before in a big study by the group in Spain, where they see that this particular variant is enriched in patients who have a very large platelets but had no effect on the platelet count. So you can imagine that the two variants can play an important role together in the phenotype of this patient. Another example of a misdiagnosis was, I found this back in literature some time ago, where they screened a patient, first a boy who had a platelet, very low platelet count of 8,000, and he died after an intracranial bleed. Parents were healthy, and they got a second child who also had a very low platelet count of 18,000, who suffered from easy bruising. So based on the clinical phenotype, the gene that you could think of was a congenital amilokaryocytic thrombocytopenia due to recessive variants in the MPL gene, but that gene turned out to be negative. And the only reason they could diagnose this patient is by performing an NGS test, where they found that Schlafen 14 had a typical missense variant, a variant that was described before, causing severe thrombocytopenia in this patient, which was not known before for this variant in Schlafen 14. And the mother actually carries the variant, but has a gonosomal mosaicism. So this is a diagnosis that could not have been made just by looking at the clinical phenotype, and without the use of an NGS panel test, this patient could not have been diagnosed. So overall, we feel that inherited thrombocytopenia and using an NGS panel test can definitely influence clinical management and is important in some decisions, and can inform actually the other clinical phenotypes that are often associated with having thrombocytopenia. You can see here that when patients have a variant in these genes, we know that they should be kept that they should be screened for the leukemic risk. Patients with GATA1 or GFI1b variants, in addition to having thrombocytopenia, often develop dyserythropoiesis or anemia. Patients with actinin or tubulin variants often have a benign form of inherited thrombocytopenia. It's known that MHH9 variants can go together with hearing loss and kidney failure. DIF1 can go together with hearing loss. This is called Aldrich syndrome. It's well known to cause serious infections, and often patients need a transplantation. And then actually patients with MPL variants and THPO variants have very deep thrombocytopenia. And actually the treatment is very different. Patients with MPL are transplanted, while THPO mutations patients with these variants can be treated with TPO mimetics. So giving some more information based on genetic testing can indeed influence management and inform clinicians about the type of inherited thrombocytopenia that's present in the patient. So today we are discussing where actually the NGS panel test should be placed in the diagnostic algorithm. Paolo Grisoli some years ago has written this up, that at this moment we now do a clinical description of the patient followed by a whole panel of lab tests that are performed, going from flow cytometry, aggregation, and so on. And actually, NGS panel test today is still at the bottom of the diagnostic algorithm. And it was proposed by him that maybe we should move this up a little bit higher, because all the lab assays are also quite expensive. And actually in Leuven at this moment, we move it up higher before doing extensive testing with electron microscopy or flow cytometry. So in patients with inherited thrombocytopenia, we usually do the NGS panel test after doing some initial assays such as aggregation and of course platelet count measurements. And we do upfront testing with an NGS panel test before doing other sophisticated assays. I just have now a few slides about more recent studies that are done outside the diagnostic environment. So this part up till now is just how we screen high-throughput sequencing for patients with inherited thrombocytopenia in a clinical environment. So what I think can be thought of when you do NGS panel tests outside an NGS panel test for diagnosis is that maybe we should do polygenic risk score for inherited thrombocytopenia. This was a paper that appeared in 2023, where they actually looked at all the pathogenic and likely pathogenic variants that have been described for inherited thrombocytopenia in patients that were recruited by the UK Biobank. So we do find variants that have been described with thrombocytopenia. And when you see in these patients, they have a lower platelet count, but there's typically carriers of, for instance, Bernard-Soulier variants. And when you see this variant, for instance, this has a high penetrance for decreasing the platelet count in carriers of this variant, and this is shown here in purple. So if you have the variant, you have a significant decrease in having a lower platelet count. Now, what is intriguing is that when we also take into account the common variants that are known to affect the platelet count, then we see that on some variants, we see that not only the rare variant has an effect on the platelet count, but also the combination of common variants. And that maybe in the future, we should implement polygenic risk scores, which altogether give a risk score for the common variants that can affect platelet count. Maybe this should be included at some moment in the diagnostic follow-up to see that on top of having a rare variant, also a polygenic risk score based on calculating different effect sizes of common variants and how this can influence the platelet count in a patient. So today in our lab, we do in the clinic the polygenic, the multigenic panel test for inherited thrombocytopenia. We have nearly 55% diagnostic rate. We do also whole genome sequencing, and we have done this over the last 10 years. And this was done in combination with different labs all over the world, and this was led by Willem Ouwehand in Cambridge. This was part of the NIH bioresource study where whole genome sequencing was used to diagnose patients with bleeding and platelet disorders. What I can say about that is that over the last 10 years, we have found several genes that cause platelet disorders, including patients with having inherited thrombocytopenia. Now, if we group in Leuven all the patients that have done an NGS panel test in a diagnostic setting, plus this whole genome sequencing effort, we still have only an additional 10% diagnostic rates by using the panel test and on top of that, the whole genome sequencing effort, meaning that still a significant portion of patients don't have a diagnosis yet. So what was successful in the whole genome sequencing effort is that we found genes that caused thrombocytopenia, and some examples are given here. And these discoveries were purely based on a statistical-based discovery using a BEVIMET approach, which is this statistical discovery panel that was developed by Ernest Thurrow, who is now in the US, but before in Cambridge. So he found an enrichment of variants in genes in DIF1 and IKZF5 in patients with thrombocytopenia. Another success story was SARC. SARC causes syndromic thrombocytopenia. This discovery was based on just having a big pedigree where different members had undergone whole genome sequencing. And then finally, another discovery was made by screening for copy number variants, where we found a big deletion on chromosome X in a patient that had part of the HDAC6 taken away. And this deletion also took away an enhancer that was very important for the regulation of GATA1, a known transcription factor that caused thrombocytopenia. And this discovery could only be made by having a copy number variant, but here you see the complexity of regulations of thrombocytopenia, where the phenotype is not only caused by having a deletion regulating low levels of GATA1, but also HDAC1, which is then causing thrombocytopenia in this patient. So what are we doing today? We're calling patients with whole genomes, and we're recalling the patients for blood cell RNA-seq. So we are capturing, at this moment we have, it's a little bit more now, but we have recruited 20, at least in Leuven controls, 9 thrombocytopenia and 18 platelet function disorders. We're taking CD4 monocytes and platelets from these patients, and we did RNA-seq of these patients. And what you see here at the bottom is the patients are indicated with the numbers, and the controls are indicated with a C number. And what you see is that in T cells, monocytes, and neutrophils, the reason why they co-segregate is based on the gender. And this co-segregation based on gender, you don't see in their platelets. There they co-segregate based on the phenotype. And you see a big group emerging here. And these are all patients that differ from patients with normal platelet count. So these are all patients with thrombocytopenia, which have a very different platelet transcriptome when compared to patients who have a normal platelet count or patients who have platelet function disorders. At this moment, we are integrating these RNA-seq data sets with whole genome sequencing data sets. And this is done in a software tool called the Reservoir, where we now see, for instance, for this patient who has microthrombocytopenia, a list of very unique variants that this patient has, but also for each of these genes, the transcript levels in platelets, neutrophils, monocytes, and CD34 T cells of this specific gene in this patient, different blood cells. So this is a way of integrating the different omics, and hopefully we find some more novel genes over time. This is my final slide. I would like to make a conclusion and the take-home messages that only for the part on the NGS panel test that can be used for patient diagnostics. So it is cheap. We do 6 months, between 4 and 6 months for each patient to do the test and to give back a result. You have to take care that for inherited thrombocytopenia, you can find genes that are risk factors for leukemia. And I think patients should be informed properly before doing the test. In addition to that, we still have some problems with variants of unknown clinical significance, which are very hard to deal with. The only way is probably to do co-segregation analysis, functional assays, and to share the variants with the community, allowing them to upgrade or downgrade these variants. And then I think the most important reason to do an NGS panel test for thrombocytopenia is that it gives a lot of information, and you can detect variants with unexpected diagnosis, as I gave you some examples, but we have more examples of that in our database. And I would like to thank all of you participating in here today, and I'm open to questions, of course. Thank you.
Thank you very much for your excellent presentation. Just a reminder that now is an excellent time to prepare questions. I see that there's a couple questions already in the chat. I will read those questions out loud, and I just wanted to put in the chat real quick, just to make sure that you're aware of the next upcoming webinar as well. So we'll start with the first question. It says, many thanks, Kathleen. This is from Dr. Fiore for your talk. Why do you use some exome sequencing rather than a panel strategy? Do you think that we will still find many other new genes associated with IT in the future? The follow-up question.
Okay. I think the exome sequencing is done in a panel setting. Maybe I wasn't clear about that, but we use exome sequencing, but we only look at the genes in a kind of virtual panel that I presented in my first slide. So the 41 genes that cause inherited thrombocytopenia. So it's not different than a panel. It's a virtual panel, and we don't look at all the exome genes, but only at the genes that are known to cause inherited thrombocytopenia. The good thing of using an exome when compared to targeted sequencing is that when there is a new gene discovered, you can open that gene in the exomes and see whether it's covered, and go back to the exomes of a patient and look at additional genes over time. And then the second questions, yes, I expect that we find new genes. Actually, one of the genes that was in my first slide is a new transcription factor, ERG, that was just discovered a few months ago. And that ERG is a transcription factor just discovered a few months ago, and is actually very similar than RUNX1 in ETV6, causing thrombocytopenia with risk for myelodysplastic syndromes.
Great. Thank you very much for the question and the answer. You have another question. It says, thank you very much, Kathleen, for this very useful talk. What would be the role of long-read sequencing in this scope?
Yeah. So I think the long-read sequencing, probably there is still room for long-read sequencing when you want to pick up complex rearrangements. So it's still a problem in exome, but also in genome sequencing to pick up complex rearrangements like inversions. We don't have any problem with duplications, gene duplications or deletions, but I think for the patients where you're very confident that they have inherited thrombocytopenia because of an effect in the known gene, I would sequence them with long-read sequencing. In our center, we don't take it in long-read sequencing within the diagnostic route because it's too expensive to do an exome and long-read sequencing. But on specific cases, it might be worthwhile to do long-read sequencing at the individual level, not for all the patients.
Great, thank you for that answer and the question. Once again, this is an excellent opportunity, just considering the level of expertise, the coordinators that are present, and the numerous health professionals that are here today. So it'd be great if there were additional questions given the time, if there are any out there. And also, I'd just like to thank everyone for being here today. I think this is a great webinar series, and I'd like to thank the coordinators as well. I think that there's 15 of these webinars is quite impressive, and just wanted to say a quick thank you. Perhaps any additional questions or comments from the coordinators or from any of the participants? From Dr. Saultier, very clear and interesting talk. Thank you. I have another comment. Thank you very much for your excellent presentation. Another comment. Thank you very much. How do you do the platelet isolation for RNA
Yeah.
sequence?
Yeah, that's a very good question. Actually, I didn't have time to go over that, but we use a very sensitive assay. So we use, so we make platelet-rich plasma and use 2 centrifugation steps, but then we also use CD45 positive beads to take out the remaining few leukocytes that are in the platelet-rich plasma. So we do an extra purification to make sure that there is no contamination of leukocytes in this extra step. And we check afterwards with flow cytometry to make sure that the platelets are really 90% pure. So we really sure that we are sequencing only platelets and not contaminant leukocytes in this step. There will be shortly a paper coming out from the SSC, from the ISTH, who has, and it's from the Rondina group in Utah, who has actually compared different platelet isolation methods for RNA-seq, and only when you do an extra step for a control for leukocyte contamination and to take out the leukocytes with bead selection, only that can be used for platelet RNA-seq, because otherwise you will have in the total you will see the leukocyte transcriptome and not the leukocyte, not the platelet transcriptome.
Great, thank you very much. Is there any other additional questions? Looks like we have another one. Thank you very much for your great presentation. Which approach is recommendable to detect structural variants in IT?
Well, so as I told you, the lesions and the duplications we just pick up, but we always validate them by MMPA to see whether they're real or not, because there's still some false positives when you do the exome sequencing. For the structural, so the insertions, and I think probably long read sequencing, but at this moment we don't do it because, yeah, we feel that we are not losing too much information yet. So in the project where we have done whole genome sequencing of patients, we have picked a few inherited thrombocytopenia patients where we were thought they were MHH9 positive, so they fit within MHH9 syndromic phenotype, and we did long read sequencing on these patients, and we didn't find any structural variants. So at this moment, I'm not too confident, or I'm not convinced that we lose too many structural variants, specifically in this group of patients.
Great, thank you both for the question and the answer. There are still a few minutes if there are any additional questions. Also, I'd like just to take a moment and thank everyone, and also to note that it would be great if people shared the webinars as well. I mean, given the level of expertise and technicality, I think it would be great. I think it's a phenomenal resource, and I think the turnout has been excellent, but given how resourceful this is, it'd be great to make the webinars more visible as well. So thank you. I have another question. RNA platelet isolation still is a problem if several
No, it's not actually. We were very much surprised that, so we had a few patients who really had macrothrombocytopenia, very low platelet counts. So by making the PRP, we use a very low centrifugation, dense speed centrifugation. But then afterwards, by the CD34, the CD45 isolation, there were sufficient platelets, even if they have very, very low platelet counts, to do RNA-seq on. So these were patients with counts below 10,000 platelets per microliter. They had very big platelets, and we still could obtain sufficient highly pure platelets for RNA-seq. We do it on children. So we have done isolations with 30 milliliters of blood. That's the minimum, I think.
Great. And perhaps there's time for one last question, if there's any questions. Perhaps wait one more minute. And if not, I would like to thank you just for an excellent presentation and for your presence. And I would like to thank the coordinators for having organized all of this. So I think with that, we will wrap up the presentation. And I thank you once again, Doctor.
Yeah, thank you very much. Thanks all for joining me here today.